Right, so we've got a midterm. It is the next class. It is Monday morning at 10 o'clock. So it's going to include all the material from the very first class until this class, including this class. There'll be 8 short answer questions, and you're going to choose, you'll choose 8 questions from 10. There'll be 10 on the test. You're going to choose 8 of them. So I gave you these examples last time. Let me give you an example of what I think a good answer to the first one would be. So explain what a paradigm is and how it relates to noticing. So first I'm going to set about defining the key terms here. So a paradigm is a set of beliefs within a science about what counts as relevant or irrelevant observations, which methods are reliable, what sort of experiments are worth running, and how evidence should be evaluated. And then I define say what we mean by noticing. So Grinnell talks about noticing in terms of what aspects of the world we should pay attention to. And then I connect the two together. So I've defined my 2 terms and then I explain how they're related. Paradigms help narrow scientists' attention down to some small set of aspects of the world. This helps solve the problem of noticing. And I mentioned that it could also lead to some things going unnoticed. Yeah, so that's about the length of an answer that I'm looking for. Not much longer than that. So when you're looking at the tests, I'll have them printed out all sort of like a question and then a bunch of sort of lines for you to write under the question in. If you get to the end of the lines, you've probably written enough. You don't necessarily have to fill all of the lines for your answer. An answer could end up being shorter than that, but I'm not looking for like page-long answers to these questions. Yeah, you could write a page about this question. You could go on and on and on. But this would be full marks for this answer, okay? So, does that seem reasonable? There's 8 of them, so you're gonna have to write, I mean, what's 50 divided by 8? Like, yeah, whatever that is, that's how many minutes you have for each of these things, yeah? 50 divided by 8. 48, 6, a little more than 6 minutes per question. So not tons of time, frankly. So, yeah. I mean, if you've got examples, those are helpful. So no extra points, but what I'm looking for is evidence that you understand what you're talking about. Anything that does that works. So an example shows me that you've able to connect something I talked about in class to the concept being discussed. That's good evidence that you know what the thing is. So a lot of these are going to have the word explain in them somewhere. A couple ways to explain things. One of them is to give examples. So if you want to say, explain to me how light works, you'd say, well, here's an example of that, how that works, and you sort of like explain the example, or like tell me about the examples. That's one way of explaining things. Another way of explaining things is to connect up ideas. So connect the notion of paradigms with, so what I've done is connect it to a bunch of other ideas, that sort of thing. Does that make sense? Okay. So I haven't filled out answers to the other 4 sample questions. Do you want to do these together? We'll brainstorm what answers to these might look like. So explain the rat park example and how it relates to the idea of paradigms. Somebody want to tackle the first half of that? So explain the rat park example. Yeah, go.
Okay, so it was where you had tests being done on rats, where you had water that was laced with heroin and water that was not laced with heroin.
Mm-hmm.
And they found that when the rats would tend to just drink the heroin-laced water until they died.
Okay.
And they just assumed that they used that as a model for addiction, and then assumed that the same principle would apply to humans, and they failed to account how the environmental conditions that existed during the test would affect the behavior of the rats.
Okay, so in addictions research, what should I say? It's got to be short enough that you can write it in like a few lines. Yeah, yeah. So yeah. I mean, you got it, but like yeah.
Yeah. Could you just say like that they had two kind of environments? One was the metal bar, and in that situation, the rat had nothing to do but just consume the heroin until they died. And then they put them in a social environment, and in that situation, the rat like thought it was more important to be able to socially interact than to consume the heroin, so they wouldn't have as much.
So yeah, bare metal cages with nothing to do but consume, I think it was morphine, and another where they had a rich social environment. In the rich social environment, rats tended to use non-fatal amounts of morphine, but in the cages, they did it until they died. That would be plenty for that. Yeah? Okay, so that's the first half of that. And now, what does that have to do with paradigms? How about, yeah.
So this is an example of like, or it relates to paradigms because the paradigm at the time was that it didn't really matter what the environment of a rat was, and that like they would react to the morphine with wastewater the same way, or that way regardless.
Yeah.
But then the researcher who developed the rat part kind of thought outside of the paradigm and then thought about the socializing of the rat, which led us to realize that your environment has a big impact on addiction.
Showed that environment has a big impact on addiction, which expanded the paradigm?
Yeah.
Good. Okay, that's a very thorough answer. That's full points. Yeah? Okay, good. Questions about that? All right, let's do another. Explain Zizek's notion of ideology and how it relates to science. So these are the questions that I didn't put on the test. I hope the questions on the test are sort of more straightforward than these, but yeah. Okay, so anybody remember what Zizek said about this? Yeah.
Was this the thing about the umpire calling balls?
Nope.
Okay.
Yeah.
Assumptions you're not aware of.
Yeah. Zizek says that ideology is the assumptions you're not aware of. The unknown knowns. Yeah. How does it relate to science? So this is a wide open question. I talked a lot a week ago about ideology in science. So you might talk about, these unknown knowns can play a role in how we frame and interpret scientific experiments, observations, let's say. For example, Marxist ideology made Lysenkoism seem more plausible than it actually was. Might be a good idea to talk a little bit about why and how that's the case. So Lysenkoism is the idea that environment matters more than genetics in agriculture, which is also a tenet of Marxism. Something like that. Okay, I think this is a particularly tricky one. Again, this is why I didn't put it on the test. Okay, one more. Here's a reasonably straightforward one. I hope that this one is doable. Everybody remember Plato's paradox of discovery? Yeah.
The paradox was that because you don't know what you don't know, that it would be impossible to learn anything. Maybe that's not the best way of saying it.
So how about, was that you can never learn anything because, why did he think you can never learn anything? Yeah. Oh, sorry. Yeah.
Oh, because if you already know something, then you can't, like you already know it, so you can't search for something you already know.
Yeah.
And then if you don't know something, you don't know what you're looking for, so you can't try and find it.
Good. That's perfect. Yeah. Yeah.
Did you say like learning new stuff was just us remembering stuff we forgot?
That's right. Very weird view of knowledge. And then Grinnell offers us a way out of this. He thinks that this is not a real paradox, that we can in fact learn things. What was it? Yeah. Yeah.
He said that like learning isn't an all or nothing affair, so like we might have ideas, but we can slowly, I guess, I don't know the word.
Like,
Make progress on learning.
excellent. If you want to really round this out, you could use the example I gave in class of dark matter, a label for something we don't understand. Having labeled it, we can develop our understanding. Perfect. Good. So those are really, really good answers. Those would be full marks answers. Yeah. Is that okay? I mean, do you accept it for the purposes of getting on with our lives? Yeah. Okay. Okay. Good. Okay. So, yeah, that's what we'll be doing bright and early Monday morning. Come full of knowledge and caffeine. No. Okay. So if there's any other questions about the midterm stuff? Okay, good. So today we're going to finish off talking about Grinnell. This is the last we're going to talk about him. Next week after the midterm, we're going to start shifting over to kind of more traditional philosophy of science stuff. So far we've been working with this Grinnell text, and it's been very much like from a scientist's perspective. We're going to do some more standard philosophy of science stuff. So we're going to start in with Karl Popper and his attempts to distinguish science from pseudoscience. We had a lovely discussion about that we had to kind of cut off in the first class, or else we would have gone on and on and on about it. So I'm looking forward to that. But yeah, so today we're going to finish off talking about credibility. So here's Grinnell's sort of expanded picture of the credibility process that we started talking about last time. The discovery claim and the credibility process. The discovery claim where you go from being an individual researcher to having written something to giving it to some journal, and that journal either publishes it or doesn't. Once it's published, it's either cited or not, which is of course the lifeblood of any researcher's career, is being not just getting published but getting cited, because that means that something that you said changed the discussion. That's kind of what people judge their success on. That's how your people judge their own success. It's also how other people judge your success. Like, did you contribute something that other people found important and interesting enough to respond to? And then it goes out to kind of the broader research community and the public at large. That's the sort of long chain of the credibility developing process. So we talked about, way back when we were doing the kind of overview of this stuff, we talked about the sort of transition from lab notes to journal article. Grinnell says this, he says, writing a paper is to substitute order for the disorder and agitation that animate life in the laboratory, to replace the real order of events and discoveries by what appears as the logical order, the one that should have been followed if the conclusions were known from the start. So I think we talked about this enough, to sort of the cleaning up of the process where, if you have a bunch of experiments that just went nowhere at all, you don't really add those, you don't put the we spent 3 weeks messing around with the setup and nothing worked and we just failed and failed and failed. That doesn't make it into the journal article. You start the story from when you hit on the working procedure and tell them what you did when it eventually worked. We talked last time about peer review. So I'll just sort of say, you send it to the editors and they either reject it without even sending it out for peer review, or if it's good enough, if it looks kind of plausible, they send it to 2 or 3 reviewers. Those reviewers are hopefully experts in the paper. They hopefully don't know you, but not so expert. So here's the balance you got to hit. They got to be people who know what this sub field you're working on is on about, but hopefully not people that know you so well that they can immediately figure out who you are, because it's supposed to be anonymous. It's supposed to be that the reviewers don't know who you are and you don't know who your reviewers are. That's to avoid, I mean, and there's obviously the threat here of petty interpersonal rivalries. Like some scientist looks like a kid that bullied you in elementary school, so you reject everything they submit. Like that's obviously would distort the scientific process. You don't want that happening. So blinded review is the goal. It's in the age of Google, it's harder and harder to get perfectly blind review, especially since lots of scientists, before their journal articles are published, they put them up on preprint archives. So there's lots of wonderful archives on the net that are not peer reviewed that you can just put your journal article up on. So if somebody just Googles the title of your paper, it often shows up on the preprint article with your name attached. So blind review, it's not perfect, but it's better than nothing. And then the reviewers say it's either good enough or not good enough. They might make you rewrite something or actually redo some of your work. And then if it gets through that, then you're published. Hooray. So that was all stuff from last time. Here's a complicated graph. This kind of web of interdependency between in the credibility process where, so you've got a principal investigator, this person who's deciding which experiments to run, how to run them, that kind of thing. So the person running a laboratory, and they're hopefully generating research papers. And those research papers hopefully justify getting research grants. And then those grants, of course, make it possible to do more research papers. So there's, the production of papers generates grants that runs the production of papers. The principal investigator runs the research paper, obviously through journal editors and reviewers. And these research papers are also looked at by the grant reviewers, the people who decide whether you should get money to run new experiments. Of course, people outside of academia are often shocked that you don't get any money for writing a journal article. Zero money. Like you don't get paid you do all this work to produce the research paper and you get zero money for that. The predatory journals will make you pay. Don't ever publish in a place that costs you money to pay for it. But yeah, and then of course off to the side, the grant reviewers are also looking to see whether your spending money on you will help also to bring up new people in the process. So getting the research grant requires the principal investigator to have a bunch of scientific credibility, that is to have a record of publication. And part of what you get a research grant for is that you demonstrate that you've got a whole bunch of people working in your lab who will gain some credibility through this process. Yeah?
Also a little bit off topic.
Okay.
I find it, especially because the authors don't get paid, I find it a little ridiculous how much they charge for you to be able to read one article.
Yes. Yes, sometimes hundreds, like $100 to just download one article. Yeah. The publishing industry is one of the most profitable rackets going right now. They make like 50% profit on investment. It's remarkable how much cash they're making. Yeah.
But there are more and more efforts to bypass the publishing industry.
Yeah.
So like,
Yeah.
in theory, now with the
So
internet, you could just have researchers just publishing their work, and you could eliminate the publishers altogether.
Yeah, okay, so that's the preprint archive sort of runs that, but then it's really hard to do the credibility process. So you need, in some sense, gatekeep. Like if it's just people putting out work and there's no peer review, how do you separate the good stuff from the bad stuff?
Well, you could add the peer review on after the fact.
Yeah, I suppose.
So everything gets published, and then people start evaluating its quality.
Right. So this is the imaginings that we're sort of saying, like maybe someday, like maybe we should have a totally different system than the one we've got right now. I'm certainly not against that idea, but we don't have another system to just easily jump to. We don't have a peer, so like, yeah.
But what about Wikipedia?
What about it?
Well, okay, so because we live in a capitalist society, there's a massive amount of emphasis put on individual ownership of ideas. In the case of Wikipedia, it's not, there's no one person who owns a particular article because you have
and there's
a synthesis.
And there's nobody giving them grants to run $1 million experiments either.
That's true.
So I'm just okay. I would love to speculate at length about a different way we could run this,
Okay,
but
fair enough.
currently there is not a different way. Currently, this is the way that science is run. Yeah. So, and certainly we'll talk about some issues that this system has. Okay, we talked last time a little bit about journal prestige. So one of the things that the grant reviewers are looking at when they're deciding whether to give you money to run your next experiment is your research papers. And the research papers can't just, so this is one of the things that is missing from a kind of journal-less thing. One of the things they're looking at is how fancy a journal did you get your article in? So the top journals like Science and Nature are incredibly prestigious, at least partly because they have a very high H factor. Impact factor. H factor is how often you as an individual are cited. The impact factor of a journal is how many times on average is a journal article in that journal cited by other people. So it can be in the 1s or 2s, it can be in the 50s to 70s kind of thing. So like in the most prestigious journals, the average article is cited 50 times or something like that. So that's a metric, it's supposed to be a metric of how much of an impact that journal has on further discussions in the science. So the fancier and more prestigious a journal you get your article in, the more it's worth when grant reviewers are looking at whether you should get more money. Yeah.
Isn't it also possible to get you cited, like, let's say 100 times by 100 different articles, but that all saying, like, this is crap?
That's possible. It very rarely happens. So I can't think of the last time. I mean, it happens some of the time, but it's actually very rare for somebody to cite another paper negatively. Like you certainly can't get an entire journal article published that just says, I read this paper and I think it's garbage. Like that just doesn't get published. And sort of failures to replicate, things like, I mean, the worst thing that can happen to your paper is it gets retracted from the journal. So if somebody in kind of the informal networks of science points out that there's serious enough flaws in the paper that it should never have been published, you can get a correction or a retraction. But that doesn't show up in these metrics. And it doesn't constitute it's certainly not the case that 100 people will cite you. And this happens in philosophy all the time, however. So philosophers can get cited for being very wrong. Wrong in some interesting or important way. Scientists almost never get cited for that. They just don't talk about you anymore. So being ignored in some sense is the worst thing that can happen to you as a career scientist. Yeah, so they don't cite you to talk bad about you, they just don't cite you at all. Yeah, which is another part of creating the kind of facade of a linear model of science. The image of steady accumulation of knowledge is at least partly maintained by this overwhelming tendency to cite only those things you consider good science and just ignore the things you consider bad science. So, okay. I think that's as far as we got last time. But of course, getting published it's great, but it's not the end of the credibility process. And I would insist that just having one, even a statistically significant result with a large effect size shouldn't be considered conclusive evidence that something is true. So one study shows that something is maybe true. One study cannot show that something is definitely true. So this is why the credibility process doesn't end at publication. So even if it's made it through peer review, there's a long road to get from there to A, being cited, and B, getting into a textbook. So here's one of my favorite examples, reasons why a statistically significant result shouldn't be grounds for concluding too much. So this is an image from the paper Neural Correlates of the Interspecies Perspective Taking in Postmortem Atlantic Salmon. That is to say, they took a dead salmon and put it in an fMRI machine and then showed it pictures of people making various emotions and asked it questions about what it thought the people were feeling. This is a dead salmon. Okay, so they were really just doing this to calibrate their techniques. This was not a serious attempt at doing a scientific study. Nobody believed that the dead salmon had strong opinions about which emotions it was seeing. They did it because they thought it was funny. However, you see these red dots here. So those red dots are areas where, using normal statistical techniques, the standard techniques in the field, they showed statistically significant activity in this dead salmon's brain in response to the pictures. Yeah? So the problem, one of the problems with, and they published this as a poster. It got talked about quite widely. One of the problems with analyzing fMRI data, particularly right now, is each one of these pixels, or voxels they call them, each one of these little areas of the minimum resolution is in some sense an independent test, right? Something statistically significant could show up in every one of those little dots. And in every one of those little, so statistical significance, you recall, says there's less than a 1 in 20 chance that something showed up by accident. But suppose you're doing 100,000 tests. Well, 1 in 20 chance across 100,000 is actually quite a good chance that something's going to crop up, right? Or just you run 100 tests, there's a 5 to 1 chance that you're going to get some statistical significant information. Is that about right? Like, the larger the data set is that you're working with, the more data that you're trolling through for patterns, the more likely some patterns are going to crop up. And that's what happened here. So this is how they explain this result. fMRI produces huge amounts of data, and the chances that the more data you've got, the more likely it is that there's going to be some signal in the noise just by accident. So that's why this is neural correlates of interspecies perspective taking in the postmortem Atlantic salmon, an argument for proper multiple comparisons correction. So basically they're making an argument for improving the statistical techniques that people use standardly, because the normal statistical techniques showed a signal where we're fairly confident there shouldn't be one. Yeah? Yeah?
But isn't like the ratio of significance to unsignificance still the same? So it's not a scientific error, it's a choice error. The scientist is choosing to show that significant result, even though, like, reasonably they should say, okay, this significance here is probably not true because of this ratio.
So, yeah, I mean, it's not, well, even in an fMRI, so suppose this was, forget for a second that this is a dead salmon. Suppose these are regions in the salmon's brain. You wouldn't expect a pattern in its gills down here or in its jaws up here, right? So if this dead salmon was responding to pictures of people's facial expression, you'd expect there to be just a few areas of significant activity. So they found significant activity. They happened to find it around where the brain was. So it's not that, if there was a real signal, we would see it in every voxel. It's, a signal can be just a few little voxels. So you can't say like, well, we found no signal in 99% of these, so therefore there's a 99% chance that there's no signal. The signal that you'd expect to find is actually very constrained. So, but I mean, the basic point that this shows, I think the basic point that you're making is right, that what this shows is not that the science is bad. It's that this is the wrong, what we got are the wrong statistical techniques. So, and that's the point that they're making in this paper is, if you use the wrong statistical techniques, you'll find signals where there aren't any. Which is again why you know, one experiment shouldn't be taken to be evidence of anything in particular. Yeah.
There's some animals that have massive amounts of nerve cells in other parts of their body, to
Uh-huh.
the point where they have cognitive
I
processing.
feel like this isn't going to be super relevant.
It's a little relevant.
Okay.
So, like,
Okay.
if you
So
did-
not super relevant.
But if you did that scan of a squid, you would see the signals lighting up on their tentacles because they basically have brain cells in their tentacles.
Okay, it's not relevant. Okay. Okay. Yeah. Yeah.
So like this experiment wasn't actually trying to find things out about like the salmon, it was trying to find things out about the statistics.
That's so when they were actually doing this, they were just calibrating their methods. This was not meant to be a serious experiment.
Right.
They were just sort of like, we'll just shove something in there. We'll run some tests to sort of calibrate our instruments, calibrate our techniques. But using a fairly I have to take, I don't do fMRI work, so I don't actually know the details of their statistical methods, but they report that using fairly standard statistical methods, they found what should count as a signal in this joke experiment. Yeah. Okay. So, just to say, one experiment with statistical significance shouldn't be counted as, we're definitely sure that this is a real thing. Okay. So what do we need? We need something like meta-analysis, right? So a scientific study, one scientific study is not that's not what we build textbook knowledge on. It's not what you sort of use as your metric for having reliable information. The standard, the gold standard, the sort of like the best current technique is to do a meta-analysis. So somebody, after dozens and dozens of scientific studies have been published on something, somebody goes through the literature and collects that data. They collect the data from the various studies and they use statistical techniques to combine them and to see whether there's a signal across many different studies. So this is currently the standard technique for trying to weed out the signal from the noise. Yeah? But, of course, just with single studies, there's a lot of choices that you have to make when you're doing a meta-analysis. So, for example, how should studies with different methodology be compared? And it's almost never that you'll have a whole bunch of studies with identical methodologies, right? The exact thing that they're studying shifts slightly. The exact way that they study shifts slightly. So these studies, in this picture, it would be lovely if it was just like this picture. Like there's just a whole bunch of identical looking graphs that all measure the same thing, and then we just combine those graphs together into metagraphs, build a graph of the graphs. That would be lovely if that's how it actually worked. That's usually not how it works. So you've got people doing slightly different types of tests. They've run slightly different analyses on it. And the meta-studier has to figure out what the best way of bringing them together is. So how different can these studies be before you're not allowed to group their data in together as though it's testing the same thing? And that's just an open question. It's a serious scientific question. It's a serious question about scientific methodology to which there is not an answer handed down from heaven. There's not just one obvious answer to the how we should make those decisions, and it's probably going to depend on what you're studying and why. So here's one of the issues with meta studies. It's called the file drawer effect. Have you heard of this before? Hands up if you've heard of the file drawer effect before. Okay, not most of you. Okay. Sorry, I have to do those little tests to see if I'm condescending you or telling you stuff that you definitely know before. So the file drawer effect is a serious worry in especially doing meta studies. So when you're doing a meta study, what you're doing is looking through the published data. But as I've said, negative results typically don't get published because what you're trying to do is trying to get your journal into your article into the most prestigious journal you can. And they judge the quality of your submission by how novel it is, how interesting it is. So they don't publish replications, and they generally don't publish negative results. So I mean, you can imagine yourself as a journal editor. You're at the fanciest journal there is, and you get a paper that said, we tried a bunch of stuff and none of it worked. Like, does that strike you as worth edging out somebody else who did find something? This is the choice that the editors are making. They got a pile of papers that claim to have found something fantastic, something interesting and new, something that might tell us about the structure of reality. And then they've got your journal article that says, yeah, we didn't find much at all. Not much happened, no interesting signals. I can tell you that this thing didn't work. That's probably not going to make it. And it's very, very rare for studies that don't work to get published. There are some very rare circumstances, but it's much, much less likely than studies that do produce statistically significant results with some kind of effect size. So, and from the scientist's perspective, you can also see how this kind of makes sense. You tried a bunch of stuff and nothing worked. Does that mean that there's nothing there, or that you didn't do the experiment very well? How do you tell those two things apart? Given that you don't know, I mean, again, we're not testing textbook knowledge. We're not going out and saying, I know how this mechanism actually operates. Now let's run an experiment that shows that that mechanism exists. You're on the cutting edge. You're trying to push forward the boundaries of knowledge. You don't know what's out there. So the fact that you failed to find it isn't really evidence that it's there or not there. Could be that you ran your experiment badly. Could be that one of your postdocs showed up hungover and spilled a beaker or something. Could be that you just aren't looking in the right place, or your reagents aren't what they were marked as. There's a million reasons why an experiment might not show something, and you don't know which one it is. So you're very unlikely to run to a journal and say, I can tell for sure that there's nothing here, because you don't know that there's nothing there. So you put it in your file drawer. You put it in your, you put your notes in a cabinet somewhere. You might tell your friends about it, so you might talk about it to other scientists. So it's not that this stuff never gets out, but it's not the case that it gets into the literature. Yeah?
So would it be worth it for like the government to specifically allocate money towards like negative results? So they would create a specific journal that only publishes negative results?
Yeah, I mean, that seems like that is one of the sort of strategies that people have been talking about for dealing with this kind of thing.
Okay.
Yeah.
I was just going to give an example of kind of one that was like published of negative data.
Oh, that'd be great.
So there was, I think it's the mRNA vaccine or something like that causes autism. There was this big.
Oh yeah, mumps, rubella, and influenza. Yeah.
Yeah, something like that. That it
Right.
causes autism or something like that. And then
Yeah.
there was a lot of papers published afterwards
Right.
that said no, it doesn't.
Right. That's a nice example of, because this, so those are the kind of conditions that it needs to be for negative results to be published. So, so somebody got into a journal the claim that there's some correlation between vaccines and autism, and it made a humongous splash well beyond where you might expect it to, and then it got way into the popular culture. So now there's a burning interest in this purported correlation. So now there's been study after study after study, and they very regularly showed no correlation. Yeah, good. Thanks. Yeah.
I think that like there's a difference in science to like publishing negative results or like publishing to like prove something wrong.
Mm-hmm.
So like I feel like publishing to prove something wrong does happen a lot, but just like a failed experiment, that's like those are like kind of two different things.
Right. So in some sense that those vaccines and autism studies are showing their hypothesis is there's no correlation, and then they go out and prove that there's no correlation. So it's not like they were trying to show a correlation, they failed, and they don't know why. What they were trying to do is show that they're, test the degree of correlation, they find 0, and it gets published. So there's been studies on this. So here's a definition of the file drawer effect. A publication bias exists if the probability that a study reaches the literature and is thus available for combined analysis depends on the results of the study. So anytime the results make a difference to whether you get published or not, you're going to have a biased reporting of the sort of like available information. And there is such a thing. I mean, I hope that it's partly why I've been sort of talking a lot about the incentive structure in publishing. There's kind of unquestionably this type of bias in what gets published. And the meta studies are not looking at the raw data of all of the scientists in their field. They're looking at what gets published. So here's a fun example. So in 2011, this was exciting times on in the philosophy of science. Daryl Bem published this paper called "Feeling the Future." So Bem is a reputable psychology researcher, but for years and years, he had this kind of secret side project of testing whether some effects could be time reversed. That is to say, so there's lots of experiments in psychology like priming. So you show somebody a picture of a fire truck, they're more likely to produce the word fire truck later. Or you show somebody a really disturbing image, they're more likely to produce negative sounding words later. So you prime them to act in some way. Loads of experiments showing this. What Bem did was have them say the words and then show them the image. And what he found was statistically significant correlations between the priming effect, which came after the behavior. So his conclusion, so in 9 experiments involving more than 1,000 participants, the test for retroactive influence by time reversing well-established psychological effects so that the individual's responses are obtained before the putatively causal stimulus events occur. Bem's conclusion was people have psychic powers, they can see the future. They are unconsciously responding to events that happen in the future. Not that they consciously knew what was going to happen, but these things like priming effects are not conscious, right? It's not like you know that you got, because you can show somebody a picture of a fire truck for 1/10 of a second, and if you do a masking stimulus right after, so you show them you flash a picture of a fire truck and then show them the bright light, they're not consciously aware that they've seen the picture, but they're more likely to say the word fire truck afterwards. Yeah? So you don't have to be conscious for priming effects to work. His claim was that he had shown, using extremely standard experimental techniques, with the only exception that he reversed stimulus and response. So the stimulus comes after the response. Using extremely standard statistical techniques, the kind of data collection and measurement that was very standard in psychology at the time, across 9 experiments, he claimed to show that people can see into the future. So there's 2 ways you could take this experiment. Yeah.
Was this debunked, or was it? Is this still?
No, I'm telling you, ESP is real. No, of course it was debunked. Of course it was debunked. Okay, so no, of course, yeah. Yes, of course it was.
That's disappointing.
I'm sorry to let you down, but
You had my hopes up.
I know. Well, I mean, here's some really good scientific evidence for it. It's as good as anything in psychology, which was horrifying to people in psychology. They got really, so this in some sense kicks off what's called the replication crisis. I think we talked about this in the first week or so, where people saw this and they did not conclude that ESP is real. They concluded that the standard techniques that he was employing, and this gets published in a very prestigious journal. This is not a joke journal. This is a serious journal of psychology. The reason he got it published in a serious journal was because it was, by the standards of psychology at the time, immaculate methodology. Yeah.
So did he believe it, or he was trying to make a point?
Oh no, he believed it. He believed it. Yeah. So these experiments were done over decades. He was paying subjects and research assistants out of pocket, so he's like using his own cash. He's kind of doing it in secret, and yeah. So he's a full-on believer, and he thinks that this is the conclusion. He's finally coming out with his conclusive evidence that this is a real thing. And people did not respond the way he was hoping. He was hoping they'd be all like, oh, ESP is real. Let's throw a bunch of grant money at this and prove that this is a real thing. People responded by going, oh no, psychology is broken. Our methods don't work. If you can get this result, that proves that our methods are not working. Yeah. So yeah, people get deeply, deeply worried by this. Okay, so, and this is relevant to what the file drawer effect that I was just talking about. Here's a little quote from an article, an interview with him. It's a really good interview. So he says, this is the guy who runs these experiments. He says, I would start one experiment, and if it just wasn't going anywhere, I'd abandon it and restart it with changes. Bem told me recently. Some of these changes were reported in the article, others weren't. I didn't keep close track of which ones I had discarded and which ones I hadn't, he said. So he's doing this over decades. He's running experiment after experiment after experiment. And if it doesn't look like his experiment is quote unquote working, he gives up on it. Yeah.
Isn't that like those other guys stuff that we were talking about?
That's right. That's the guys who, yeah. So it's the same thing that they were doing when they discovered the relationship between DNA, messenger RNA, and proteins. Yeah, so this should remind you of what they were saying about that. And again, I really don't think the lesson should be that DNA doesn't get transcribed into messenger RNA into proteins. Like that one I'm willing to take to the bank. This one, not so much. But it's hard to say exactly what the difference is, isn't it? One difference is that the DNA to proteins thing, it's not just that they discovered a statistical relationship between these things, they discovered a mechanism. So they discovered the causal physical relationship between. So there's, you can say which chemical it is that latches onto the DNA and transcribes it into the messenger RNA. You can say which chemical it is the messenger RNA gets fed into in order to get transcribed into proteins. So it's not just a pattern of data, it's a causal mechanical relationship that gets identified. Whereas Bem doesn't do that. So he's not saying, he has no thesis about how ESP works, especially no thesis about how it doesn't violate the laws of physics. He's just saying, look, I found a statistical pattern. And yeah.
I don't know if this is right, but something also that is like differentiates between the two is that like there's nothing proving like how DNA transcribes mRNA to protein like wrong, but like it's proven wrong. There's no one has been able to prove that that's wrong, but like this is like you are able to prove this is wrong.
Yeah. I mean that is a big difference. But how do you, I guess the question is, how do you know that in advance?
Right.
So as soon as this gets published, people look at it and go, that can't be right. They immediately sort of reject the conclusion and start questioning the methods that produced it. Whereas with the kind of DNA to protein thing, it's not so clear that there is such a route, or at least not a straightforward route. So people did attempt to replicate these things. No signals were found. No clear evidence of this was understood. And people to this day don't believe in, well, at least the scientific community doesn't accept the conclusion. And the suspicion is, he sort of admitted to us exactly what happened here. That is, to hit statistical significance, you have to show that there's less than a 1 in 20 chance that you came up with it by coincidence. So what you do, run 20 experiments. It's likely to be one that has a pattern in it, You don't publish 19 of them, and you send the one in for publication. It's very similar to the pattern of what happens with the dead salmon. Yeah? So he doesn't know, he doesn't remember how many started and failed. He doesn't know, so he didn't finish, he just stopped doing them. Yeah.
You're saying earlier with the meta-analysis is kind of when, obviously something like this, why did it make such a big splash? If there wouldn't have been a meta, obviously, because it's only one small.
Right. It made a big splash because of so again, this Journal of Personality and Social Psychology, it's a top journal in the field. It's a journal that people read, it's a journal that people respect. And the conclusion is so wild that people were immediately unwilling to credit it. However, what made this sort of make a big splash is the uncomfortable tension between the wildness of his conclusion and how immaculate his methods were. So his methods reached all of the standards in the discipline of what counts as good science. And they never have published it if it hadn't, right? If there was any problem with his reported methods, they would have immediately rejected it. There's no way it would have gotten through if it wasn't really, as good as the discipline can ask for. And so that, okay, here's a conclusion we know can't be right, or at least we're reasonably confident this isn't a real thing, but it was produced by methods which we're confident in. And people decided that it basically shattered people's confidence in the methods that they were using at the time. It kicks off this large-scale effort to rethink methodology in psychology, essentially, to start going back and retesting a whole bunch of what was taken to be very reliable results. Yeah.
Just out of curiosity, was this guy ever discredited or at least not taken as seriously from this point on?
I think I don't really know. I don't know what his personal life is like at this point, or where his career. Not his personal life. I don't know where his career is at this point, because he was not an insignificant researcher. He had a full rich, and he was not a new researcher either. He had a full rich career of perfectly respectable publications. It's just that their conclusions weren't that you can see the future. You don't get to be a researcher for 30 years without publishing a whole bunch of stuff that people consider credible. So he's not like a young guy. He's not somebody who had failed to attract a whole bunch of grant money. He was a respected psychology researcher. Another reason why he got published in a reasonable journal, because they're not going to publish from nobody's. Yeah.
How did this make this through the peer review process?
Well, it made it through because he's a good psychology researcher relative to the standards of the discipline, and he did everything that could be asked of him in terms of the standards at the time. So within the paradigm of psychology, there's what are considered reasonable research methods, and he checked every single box. They couldn't find anything to take exception with. So if they take exception with his methodology, they have to take exception with the whole field. And those peer reviewers didn't do that because they weren't willing to say this doesn't. So the job of a peer reviewer is does this meet the standards in my field? Not is my field a good one? It met the standards of the field, so the peer reviewers found nothing that they could say, we should reject this. Once published, so that's why the credibility process doesn't stop with journal publication. It continues after publication, so people start reflecting on, what does this mean that this got published? Okay. Yeah?
So did he intentionally publish that to show that the system was flawed?
No, he believed it. He was a true believer.
Okay.
Yeah, no, he believed that he was showing a real result. It's just that everybody else thought that this showed that the system was flawed.
Okay.
Yeah?
So sorry, I might have missed this, but so how does this relate back to the file? Are we relating this back to the file?
That's right. So I think one of the things that people use to explain this result, supposing we're rejecting the ESP hypothesis, one of the things that people use to explain this result is what he's describing here, which is a failed experiment just gets shelved, a successful one gets written up and reported. And if you do that often enough, if you start enough experiments, just by random chance, you're going to get a signal that looks like it's a real thing. So if you start 20 experiments, the chances are you're going to find one. Start 20 experiments on random numbers, chances are you're going to find one with statistically significant patterns in it. That's the lesson. So he's saying, like, look, I didn't, he's a true believer, which means that if he sees one isn't going anywhere, I would abandon it and restart it with changes. And there's no telling how many times he did that. He did that until he came up with the result he was looking for, because he believed deeply. Again, he's spending his own money to run this research. He's kind of doing it, not in secret, but like certainly not advertising that he's doing it, because he knows how people will react, kind of thing. So what do we do about this is the question. The file drawer effect, of course, if you're doing a meta-analysis, what you're looking at is the published research. You're looking at what's in the literature. You really can't go around, as just a matter of practical considerations, you can't really go around and rummage through everybody's notes and put together all of the experiments that didn't work. You can only look at, just because of how much work that would be, you can only look at what's actually being published. So that means that if something looks like a real effect, so if something is in the literature as a real effect, like, for example, ego depletion. Have you heard of this? So there was, for a long time, there was a kind of strong belief in psychology that there's a thing called ego depletion, where once you've used your willpower on something, you kind of use it up. So your ability to sort of like decide to do something is a kind of finite resource. And if you, for example, make a subject do an effortful task for 10 minutes, that depletes their power of will, and then if you test them afterwards, they'll be less able to do things that require willpower. So ego depletion. It was considered a real robust result. After the stuff by Bem comes out, people's sort of the bottom drops out of their stomach and they think, oh God, we're doing something wrong here. They start trying to replicate a bunch of the big important results from psychology, and things like ego depletion turn out to be very hard to replicate. So for like a decade, this was considered a robust result, partly because once it's in the literature as a robust result, people consider it reliable. So if you run a study on ego depletion and it doesn't show a result, you don't publish it. You run a study on ego depletion that does show a result, it goes in the literature, and then somebody doing a meta-analysis says, look at all these studies showing that this is a real thing. So even the gold standard of trying to put together huge numbers of studies into an overall picture of what the literature has shown will be subject to the file drawer effect. And if your study doesn't show something that everybody else thinks is real, it's very hard to get it published. So what do we do? Here's a possibility: pre-registration of trials. So before you start doing your experiment, you get in touch with some central agency. There's a bunch of sort of groups that are doing pre-registration of trials now. And you say, here's what I'm planning to do. Here are the methods I'm planning to use. And then you're sort of, you're locked into like doing your research in a certain way, and it encourages the reporting of negative results. You've already told these people that you're going to run this experiment. If you just ghost them, that looks really bad, right? So you're going to tell them if you didn't come up with a result. And it also, by sort of pre-registering the methods you plan to use, it mitigates another worry that we talked about, which was p-hacking. So p-hacking is where it's not just that you run the experiment over and over until you get the result you want. you run different types of analysis until it shows a statistically significant result. So this child is saying, it's easy. First you shoot the arrow, and then you take out your paintbrush, and so this is essentially the worry, right? You run an experiment, and then you decide after you've run the experiment what type of data analysis you're going to use on it, because you want to choose one of the hundreds of ways or thousands of ways that you could analyze the data. You're going to pick one that makes it have a statistically significant result. So you're going to choose which outliers to scrub out. You're going to choose how many trials to run. You're going to choose which type of statistics you're going to run on it until you find one that gives you a publishable result. And that's exactly like just painting the bullseye around the arrow that you've already shot. Yeah? So pre-registering, A, it makes sure people report negative results, and B, it makes sure that they don't do this p-hacking stuff, sort of picking and choosing their data analysis methods to fit their need to get something publishable so they can continue to get grants. So again, I'm telling you this at least partly. It all goes back to the linear model of science. The illusion that there is one scientific methodology, we've already got it worked out, and that we just turn the crank. We just sort of say, I'm going to apply this one method, and it's just going to keep working. The danger of that is that you don't realize that we're still working on it. There's never been just one scientific method. And it's not a finished product, it's a work in progress. So the fact that, so BEM's thing came out in 2011, yeah? 9 years ago. And 9 years ago, he was using methods that were completely, again, there's no way he would have gotten published if his methods weren't absolutely up to the standard of the discipline at the time. What that did, again, this is not supposed to reduce your confidence in science, although it might be worthwhile thinking about whether some specific result is worth believing. The thing that makes science amazing is that they take information like this and then rethink their methods. Yeah? So the rise of things like pre-registering results are a response to figuring out that their methods weren't good enough. We're going to try something new. We're going to try something better. We're going to continue to try to improve the methods as the evidence kind of comes in. And that openness to and sensitivity to evidence from the world, if you had to pick something, so next week after the midterm we'll talk about science and pseudoscience. If you had to pick something that's characteristic of science, that sensitivity to the results seems to be the most characteristic thing. Yeah? It's maybe not a good definition, but that's what makes science. If there's something special about science, that's it. The willingness to go back and say, oh, apparently we're doing it wrong. Now we're going to do it better. So individual studies weren't good enough. We started doing meta-studies. Meta-studies have some worrying properties, like the fact that there's a file drawer effect. The file drawer effect was identified, it was studied, it was thought about, and that leads us to something like pre-registration as a method of trying to improve the process. It's this constantly self-changing, self-improving process. So that's what's wrong with the linear model. It imagines that this is already finished. It's not finished. We continue to work on it to this day. We're going to have to continue working on it going forward. So there's been some pretty good studies comparing, for example, here's a meta-analysis. It's a comparing meta-analysis versus pre-registration. So looking at something about which there's been a meta-analysis and about which some of these large collections of pre-registered studies have been done. So looking at the same subject matter. So psychology is doing a bunch of this work. So they're big groups of researchers pre-registering their studies, trying to increase the rigor of their methodologies. So when they say, this was published in 2019, so late 2019, this was published like 2 months ago. They say, quote, "We find that meta-analytic effect sizes are significantly different from replication effect sizes for 12 out of the 15 meta-replication pairs. These differences are systematic, and on average, meta-analytic effect sizes are almost 3 times as large as the replication effect sizes. You don't know what that noise is?
It's the HVAC system.
Ah, okay. So, this should be, this result should be worrying, right? I'm mildly worried about the noise, but I'm more worried about the result. The effect size, statistical significance tells you not really whether something, it just tells you whether something is very likely to be due to chance or not. Effect size is kind of more important, right? So when you're looking at the effect size, that's how correlated are 2 variables? How strong is the correlation between 2 variables? And effect sizes being 3 times as big in meta-studies as in replication studies is very worrying for the meta-studies. So replication studies are very likely to be a better method than individual studies that are unregistered and just doing meta-studies on them because of things like the file drawer effect, right? Because of publication biases, because of p-hacking, that kind of thing. So finding that when you do a more rigorous methodology, your effect sizes are 1/3 of what they were before is chilling. I think that's chilling. But again, it's evidence that scientists are interested in and trying to improve their methods, trying to get better, trying to do things in more rigorous ways all the time. Yeah? So this is not evidence that science is broken. This is evidence that it's working. But that it's not there. It's not a finished product yet. That's the point. That it's not a thing where you can just run one study and expect to believe the results of it. Yeah.
Won't a lot of these problems be resolved as artificial intelligence is used more and more to do research? Because eventually all researchers will be replaced by artificial intelligence.
Well, you got to program the artificial intelligence somehow.
Yes.
And if we program it to do meta-analysis, then no, it wouldn't improve anything.
So you're worried that they'll program the artificial intelligence researcher in the wrong way?
Well, I'm just saying that the fact that it's a computer rather than a human doesn't solve any of the methodological questions.
Okay.
If you program an artificial intelligence to do something ridiculous, it'll do something ridiculous.
Yes.
If you program an artificial intelligence to do something smart, then it'll do something smart. Currently, there's no artificial intelligence on the planet that can decide how to do things for itself. What we've got are AIs that are very good. If you tell it what its goal is and what it's allowed to do to get to the goal, AIs are very good at connecting those 2 things. But there's none on the planet that says, you know what? I don't think meta-analyses are what we should be doing here. I'm worried about bias in the literature, and I'm going to try to do a different type of experiment. There's nothing even remotely like that going on right now. So maybe after the technological singularity, things will be better, who knows. But for the time being, I recommend that we work with what we got. We can't get there unless we work with what we got, right? No. Okay, good. Okay, so meta-analysis versus pre-registration, sort of worrying. I'd like to talk about another part of the credibility process right now. So just to round this out, because it's not always the case. So the diagram that Grinnell gives us, there's a sort of stepwise process. You go from the research, from the lab to the journal article, from the journal article to the community, and from the community to the textbook. And then from the textbook to the world. And much as I really like science news. Every day I read the news and there's something horrible going on. And every day I read science news and there's something nice. We figured out something, we solved some problem. I really enjoy it. But it is, I would like to propose to you, a dumpster fire. An epistemic dumpster fire. A kind of unmitigated catastrophe of loose and fuzzy thinking that leads to poor inferences. I summarize this on the slide. This process has some serious downsides. Yeah. Okay, so science journalists, typically, they're on the cutting edge of what's going on in science. So they go around talking to scientists, interviewing scientists, reading the most recent published research. So they're not, science journalists do not go to a textbook published in 1995 and pull out some interesting fact and report it as news, because that's not news, right? You can't do that. So what they do is do cutting edge research. But what we've been talking about all class is the problems with taking one scientific study as evidence of kind of anything. So there's a bit of risk there. Now, a really responsible science journalist who had really good editors that let them say responsible things would make careful claims about the consequences of a single study. That's not what happens. So I picked this kind of at semi-random from Science Daily, which is, I think, one of the more respectable science news outlets. I like Science Daily. I think they mostly do good work. They're certainly not wildly irresponsible, and certainly not wildly irresponsible relative to the field. But let's check some health news. Oh, okay, health news. Let's see what's going on. So we're on a reputable website. It's dedicated to science news. That's their whole jam. And we see things like breaching brain barriers to attack tumors. Oh, that sounds cool. How about beauty sleep could be real? Oh, that sounds interesting. Let's find out what beauty sleep can be real is. So a nice catchy headline, short, catchy. It's got a picture of an attractive person stretching in a very expensive looking room. Oh, my interest is through the roof. Let's find out what this is about. Okay. So shifting from the headline to the article, we find some, let's say, differences between what the headline says and what the article says. So typically writers don't, this is a fact about journalism in general, writers don't usually get to write their own headlines. The writer writes the article and the editor writes the headline. The writer is the one who maybe talked to the scientist, read the journal article, they've done some research, they know what they're talking about. The editor's job is to make you click. So that's what the title is written in terms of, and the journal article might say something slightly different. So under the beauty rest might be real, we find the following. Biologists from the University of Manchester have explained for the first time why having a good night's sleep really could prepare us for the rigors of the day ahead. Nothing about beauty anymore. We're just talking about whether it helps you. So the study in mice and published in Nature Cell Biology shows how the clock mechanism boosts our ability to maintain our bodies when we're most active. And because we know the body is less precise as we age, the discovery, argues lead author Professor Carl Cadler, may one day help unlock some of the mysteries of aging. Okay, so we've dropped the idea of beauty. We're now on to something about maintaining our bodies. And we've got this fun, here's a word that might escape, if you're reading quickly, here's a word that might escape your notice. The study in mice. This study was not about people, it was on mice. Okay, so they don't really the picture of the woman in the expensive looking room stretching doesn't really suggest that this was about mice. They should have an attractive mouse stretching in a very nice mouse apartment. So they don't really advertise that it's about mice. They hooked us with beauty because we're all interested in being attractive, but there's nothing about attractiveness here. There's something about the rigors of the day ahead. Okay, so substantial difference between the headline and what's in the article. Okay, fair enough, you gotta advertise, whatever. So now let's look at the study itself. So they link to the study, which is nice. So here's the study. Circadian control of the secretory pathway maintains collagen homeostasis. Here's from the abstract. Here we show circadian clock regulation of endoplasmic reticulum to plasma membrane pro-collagen transport by the sequential rhythmic expression of SEC61, Tango1, PDE4D, and VPS33B. The result is nocturnal pro-collagen synthesis and daytime collagen fibril assembly in mice. Okay, what happened to beauty sleep? So they did this experiment on mice in these genetically engineered mice who've been engineered to have basically broken circadian rhythms. So your circadian clock is the kind of internal mechanisms which regulate your wake sleep and let you know what time it is and all that kind of thing. So they took some mice who've had their circadian rhythms intentionally broken at a genetic level, and then they found that certain types of fibers, so the irregular profiles in the tendons were thicker, weaker, had reduced elastic modulus. So D1 fibrils, their interaction with D3 and D3 fibrils, and their regulation by the circadian clock are important determinants of tendon tissue integrity and function. So we've come a long way from beauty sleep is real, haven't we? We're talking now about the tendons in mice who have not even been sleep deprived, they've just had their circadian rhythms disrupted. So it's not that the mice didn't sleep, it's that the internal homeostatic regulators that tell you when it's time to sleep have been genetically disabled. And what they found was that their tendons were a bit thicker, a bit weaker, and a bit more brittle. Okay. Now, if they had published a Science News article saying circadian control of the secretory pathway maintains collagen homeostasis, I probably wouldn't have clicked on it, to be fair. So the editor actually did their job there. The editor produced a nice catchy headline with a picture that looks interesting. They got me to click on it, but it's been a hell of a ride. When you go through from headline to article and from article to the original research, boy, it's a journey. Boy, it's a real journey. Yeah. So, and this is, again, I picked an article I did not fish for this. I picked an article almost at random from Science Daily and just A, read the article, and then B, looked up the journal article that they were referencing as the research that backs up these claims. Yeah.
There's been a bunch of research done on the health effects of wine,
Okay.
simply because those
People love
types of
wine.
studies are more likely to be published,
Mm.
or people will take interest
Mhm.
in that sort of thing.
Mm-hmm. I've seen, so what is it, resveratrol. So there's a trace component in red wine called resveratrol, I think. It occurs in very small amounts. And if you give just boatloads of it to mice, they have a bunch of health benefits. And so they say, high concentrations of this trace ingredient in red wine has big health benefits for mice. And then the headline is, wine makes you healthy. Really, this is how the kind of information pipeline goes. And this is the dangers of going from, of kind of knowledge not going through the standard credibility process. So yeah. That's right. Yeah. So this is not something that that's exactly right. This is not sort of like a scientist doesn't go at this and say, beauty sleep could be real. So this is not where scientists go for their information. Absolutely. And I'm just trying to illustrate this as, because the credibility process doesn't end with cited journal articles, and it doesn't really end with textbooks. It gets broadcast to the larger world, and the kind of reliable credibility process goes through essentially textbooks. And this is what I'm trying to illustrate here is just what happens when you skip the whole process of, after it's published, leap straight into the public. So we've leapt over 4 steps in the credibility process here, and I just wanted to show you basically what happens when you do that. So the ways that we should treat what gets broadcast to us non-scientists as the real results of science, and it's just this bizarre kind of treatment of, I don't know, maybe there's some scraps of beauty sleep credibility here, But there's certainly nothing in this article. So I read some of the article. There's nothing in this article about trying to connect this with A, humans, or B, like looking good. Like there's just nothing in the original headline is present in the article. And it's like the further you go down the rabbit hole, the less resemblance there is between how it's sold to you for clicks and what the actual research shows. I don't know. So this is why I give you a dumpster fire. Here's my favorite, another one of my favorite science Twitter accounts. It's called Just Says in Mice. This is dedicated to retweeting science news articles. So skin cream applied to mosquito bites stop viruses infecting mice. So this one actually did say mice. Huge numbers of journal articles are just like the one I just showed you, where circadian control of the secretory pathways maintains collagen homeostasis. It's a mouse study. By the time it gets to the science news, it's about people. So the fact they don't mention the fact that this is a mouse study, not a human study. So this delightful Twitter account reminds people that in fact most of these studies are done on animal models, and that the inference from the animal model to what happens in humans is it's an inference. It's not trivial that this is going to apply to us just as well. So strong recommend for Just Says in Mice. Okay. All right. That's it for credibility. That's it for Grinnell. We'll see you at the midterm. And also, I promised I would post a list of key course terms. I'll do that tomorrow morning. Yeah? Tomorrow before noon, I will post a list of what I consider important, central, testable key course terms. All right. Thanks, everyone.